Part B structured question on Chapter 2, which is Gene Technology. Figure 3 shows the step in polymerase chain reaction or PCR. So basically PCR is a method to amplify or make many copies of specific DNA sequence in vitro or in the lab. So in other words, it is basically replication of DNA in a test tube. So in order to carry this technique or polymerase chain reaction, the tools that you require are the DNA sample, and then in order to amplify the DNA sample, you will require the thermocycler or the machine, DNA amplifier machine or the PCR machine. And the another tools that you require are the free DNTPs or free nucleotides, DNTPs, to assemble the nucleotides together to make many copies of the DNA, and then also the enzyme, which is Taq polymerase. So those are the tools required for PCR. So during PCR, it is consists of 3 basic steps, okay, that occurs in cycles. So it depends on how many cycles that you want. So the first step is denaturation basically. So denaturation occurs at a very high temperature, which is around 94 to 98 degrees. Okay, 94 to 98 degrees Celsius. So this during denaturation, basically the original sample of the DNA is exposed to high temperature, and the purpose of exposing the DNA to the high temperature is to break the hydrogen bonds between the two DNA strands. Okay, so you have hydrogen bonds between the two DNA strands, so the high temperature will break the two DNA strands so that they will become the template to synthesize the new DNA molecules, okay, new DNA molecules, to make many copies of new DNA molecules. And then for the second step is annealing. Okay, so this at this step, which involve the temperature to be lowered, okay, up to 50 to 68 degrees Celsius. Okay, so the temperature will be around 50 to 68 degrees Celsius, and during this stage, PCR primers will bind to each of the DNA template, okay, to each of the DNA strands. So these are the primer, or we call it as the PCR primers. And then for step 3 is extension, which occurs at 72 degrees Celsius. So during this stage, free DNA nucleotides that are complementary with the original strands are added by the enzyme Taq polymerase, and during this stage the temperature is risen up to 72 degrees Celsius, so that it helps to form the new DNA strand. And then at the end of this stage, it produces two identical DNA molecule. Okay, DNA molecules. So basically, for the first question, based on figure 3, identify step 1 and 3. So step 1 is denaturation. Step 3 is extension. State the significance of step 2 in PCR. So this question is worth 1 mark. So when every time a question asks for significance of something, so basically you have to give what is the importance of that topics. Okay, so basically step 2, as you know, it is annealing, and annealing is basically it is the step where primers bind to the two DNA templates, okay, the two DNA strands that have been separated. So basically the importance of step 2 is to allow primers to form hydrogen bonds with the ends of the target sequence. Okay, so it allows primers to form hydrogen bonds with the ends of the target sequence. So that is the answer for this question, question number 2, for 1 mark. What are the advantages of using PCR? Basically, for this question, it is worth 2 marks. So you have to give 2 advantages of using PCR. For the first point, basically, the advantage of using PCR is that when you are working on a DNA sample and you have a limited amount of the DNA sample to work with, okay, so it gives you the advantage of multiplying the DNA sample, the small quantity of DNA sample, to produce many copies. You can amplify the small amount of DNA to form many copies, billions of copies of that specific DNA sequence. Okay, so you can work with small quantity of DNA sample that are available. For example, if the DNA sample you obtain from, for example, from a crime scene, or the DNA samples that you obtain is from a limited quantity of samples of cells that you have, okay, so this gives you the advantage of multiplying the DNA into many copies, okay. So that those are the two advantages of using PCR. Table 1 shows different DNA samples. So this table shows you sample from sample A, sample B, and also sample C. And for sample A, it gives you the number of best pair for the sample for DNA of sample A, which is, for sample A it is 120 best pair, sample B is 200 best pair, and sample C is 50 best pair. So sketch and label the pattern of DNA fragments band in figure 4 in this figure by showing the information by using the information in table 1. So basically, when you run DNA sample, when you run jet electrophoresis, okay, so the DNA fragments will separate according to their size, okay. So the smaller DNA fragments, okay, DNA fragments has the lowest number of base pair or nucleotide, or the mass is small, so it means that the DNA sample will move faster, okay, will move faster along the gel. And then so it means that for sample C, the DNA fragments will be the furthest, okay, from the well. And then as for fragment B, because it has the most number of nucleotide pair, so it will travel the slowest. And as for sample A, so it will be around here in between, okay, which is around which is the base pair is 120 base pair. So the band, the DNA band, it will be around this region. So for the 3 marks, you have to correctly place the DNA bands, okay, on the gel. How do you determine the position of bands for the DNA fragment? So for this question, it is worth 1 mark. So one of the way you could determine the position of the DNA bands is based on the size of the DNA. Okay, so if the size of the DNA is smaller, so it means it will travel the furthest from the gel. And then if the size of the DNA fragment is the biggest, okay, so it will be nearest to the well of the gel. And then another way to determine the position of the DNA bands is through the voltage, okay, the voltage of electrical current. So it means that the higher the voltage, the more quickly the gel runs to separate the DNA fragments. And for the last question, name one type of gel that commonly used in gel electrophoresis. So there are two types of gel that you can use for gel electrophoresis, which is, the first one is agarose, and the second one is polyacrylamides. So you could answer any one of it, agarose or polyacrylamide. So this is the end of discussion for exercise on chapter 2, which is on gene technology. So if you have any question, you can refer to your lecturer. And then just to remind, for your midterm test, it will cover chapter 1 and also chapter 2. Thank you.